So thank you very much, everybody, and thank you for the very kind invitation to speak today. So what I'm going to do is hopefully just give you a bit of an overview as to how does a larger company, a pharma company, think about taking cell and gene therapies and turning them into viable products that can be, first of all, approved by a regulatory authority and then hopefully marketed, and marketed successfully, ideally. And so perhaps where this is helpful is if you are an academic or with a small company, thinking about the types of things that a pharma company will look at if you're ever thinking about partnering up. So these are my declarations, my disclaimers. I will mention a product, Strimvelis, which is the first ex vivo gene therapy to be approved. It's been on the market for a little over 18 months. Nothing I say should be considered as promotional at all. So a little bit of history about GSK in the cell and gene therapy space. We started a collaboration with the Telethon Institute of Gene Therapy and the Hospital San Rafael back in 2010. And you can see the 7 indications over there that we initially partnered with them on. And so we've worked very, very closely with them, learning a tremendous amount from them as we went along. And you can see where we are at the moment. We have 1 approved product, and then another 3 which are currently ongoing in clinical trials, and the last 3 are pre-option and still in the preclinical phase. So that's a little bit about where we are. How we've worked with them, well, most of you know, and Luigi gave a fantastic speech earlier on to show some of the incredible work that's going on there, and so we've really learnt an enormous amount in the preclinical space about how we take these programs and start turning them and thinking about moving into humans. And where GSK got involved was helping and working very closely with the Telethon Institute to really help design and operationalize these programs and then take them forward into the regulatory environment, and then obviously from a commercialization perspective. And you can see an overview of the types of things that we do and we do together. And I think it's critically important that if you as a small company are collaborating with an academic or vice versa with a large company, you think about maintaining that way of working very, very carefully and clearly. And then additionally, not forgetting about the manufacturing collaboration which we have with MolMed. So everyone here is in cell and gene therapies, and we know developing them is tough. This is a really, really challenging environment. At the moment, we're primarily going after small numbers of patients, and I'll talk in a moment about some of the challenges that we face with small numbers of patients. The pathways are new, the regulatory pathways, the regulators are just learning about these things, and we've seen some changes recently. We've seen 21st century cures come out in the US at the end of last year. We've seen the Japanese regulations which are adapting, and everyone's learning, and we're learning together, and so it's critically important that we engage with regulators early and often so that they can really understand your cool technology, and they can understand what this amazing thing is that you've developed, and you can take them along on that ride, and that will make that final discussion right near the end so much more bearable and easier. There are no adequate surrogate outcomes at the moment, and this is a real challenge, and this goes down to their no proven long-term efficacy. So, we were very fortunate with the ADA SCID program of the first patient having been treated in excess of 15 years ago, but most of the data that's coming out that's really reproducible, that's being used in clinical trials, has only got maybe 3 up to maybe 5 years worth of data. And if you're going to speak to a regulator or you're going to speak to a payer and you've only got 5 or 7 years worth of data for a product that potentially lasts the patient's lifetime, that's kind of a difficult discussion to have. So think about it really early. What are your clinically appropriate surrogates that you might use, and how are you going to show that long-term efficacy and that long-term safety? So let's look very briefly at some of the CMC challenges. Carl's given us a really great overview of the types of things to look at. I'm going to focus more on the translation, taking a manufacturing mechanism and a way of manufacturing a product from an academic institution into industry. And you need to think about that. You need to think about the comparability work that needs to happen. Think about your clinical quality attributes. What are they, and what do you need to take into account as you scale up and you scale out? And as you move into a more industrialized mechanism where you're dealing with, very often, particularly for autologous products, you're dealing with starting material that varies tremendously. How are you going to cope with that? Typical big pharma and pharmaceutical environments aren't used to a variable starting material, so you need to figure out how you'll cope with that and how will you take it forward to have a standardized product at the end, because the regulators are going to say to you, what are your release criteria and why can't you make them tighter? And so you need to think about how do you standardize things as much as possible so that you come out with that regulated product. Early manufacturing development, I think this is absolutely critical. The last thing you want to do is get into phase 3 and decide you're going to make some critical change to your manufacturing, because then you're going to have to go all the way back to the beginning unless you really understand what you're doing, what your manufacturing is, and what those critical attributes are. Automation is critically important. We need to be thinking about automation. We need to be thinking about manufacturing by design. What you will see in the academic institution is a very specific way of doing it, and very often relying on people that do this day in and day out, and they're very good with the techniques that they handle. When you move into a much larger manufacturing mechanism, you don't have those people. And so being able to standardize it and take the humans out of it and automate is far, far better. It's going to improve your reproducibility, it's going to improve your costs. And at this stage, we need to work really hard on improving costs of what we do. Logistics. We heard earlier about some of the logistics, and you take the patient, you take the cells. If you're using an autologous product, you take them to the manufacturing facility, you're manufacturing, and you give them back to the patient, and that's easy. But what happens when the patient is not near the manufacturing facility? What happens when those cells that are taken out of the patient are considered starting material? How do you check them? How do you test them? There is the advanced therapy medicinal requirements, and also some of the testing for human tissues that require cells to sit while you test them. You need to be thinking about all these types of things. Now, this is actually, interestingly enough, a simplified version of this whole mechanism, and this just simply shows the patient being treated. So you have the cells taken out, the quality done, the transport, the QC at the manufacturing environment to make sure that those cells reach the appropriate standard, so that if this check is missed over here, this may fail over here, and then all of a sudden the guys in manufacturing say, sorry, cells aren't good enough, we can't manufacture. And unfortunately, there's always a patient, and very often a sick patient on the other end of that, and you never ever want that to happen. Drug product is then manufactured, frozen, transported to the hospital, potentially stored, thawed, and the patient treated. But you also remember you need to take out fractions and samples, and these may be for QC and a variety of other tests that are required potentially for release, and wanting to know your QC product and your process that need to be tested. You also need to think about your patient and the logistics around the patient. We're putting gene therapies into these patients. We're changing their genome. We can't give them this therapy and then wave goodbye to them and watch them walk out of the clinic. They need to be brought back and monitored regularly. And we have in place a 15-year registry. So we need to find a way to bring those patients back and ensure they're circling back, and we continue to have that data coming in so that we're learning and we make sure that we make these products better each time. So this is a really complicated area that you need to think about, and you need to think, how can I simplify this as much as possible for when it moves into a full-blown commercial product? The clinical and the regulatory and the scientific challenges, I'm going to touch very, very briefly on these. Most of you will be aware that the science is moving substantially faster than we're able to develop products, and we're able to develop therapy. As I mentioned at the beginning, we have a product, Stromvelis, which is for ADA SCID. Now that uses a gamma retrovirus. Since then, people have started moving on using other vectors and things like that, but that was developed a while ago, and it works really, really well, and there's no reason to change it. But it's worth thinking about how science and how things have moved on, and that we're still, though, thinking about the vectors and what's the best vector, what's not. The preclinical development and the requirements that the regulators often have are still based on small molecule development. And so when you're looking at these things, you need to think really carefully and pragmatically about what is appropriate for your therapy. If you've got an autologous product, how are you going to show overdose? How are you going to show the challenges with overdosing a patient on their own cells? If you're giving into a very specific area, how are you going to show genotoxicity? How are you going to show reproductive toxicity? So think about all of these aspects and how you're going to address them with the regulators. Clinical trials are really not advancing fast enough. We're having a lot of challenges. We're still using the same old methodologies that we always have. There are a variety of new mechanisms that the regulators are trying to bring in, but very often they're just not serving the purposes that we need. So we need to be thinking about how do we do things better? How do we move into short trials that will predict longer trials so that we can get these therapies out to patients and make them available to patients sooner? So when we went to the regulators, we had some really, really interesting discussions, being one of the first therapies, and these were some of them. So the first one was, they sat down and said, right, what is the vector? Is the vector a starting material? Is it part of your manufacturing? Or is it actually a drug product? So how do you classify it? And so this was very, very interesting, and what we've actually found is the EMA consider it very differently to the FDA. So the FDA, they actually consider it as a drug product, whereas the EMA will look at it as starting material. But the fact remains, you need to understand not only your product, but even the vector, even if you're getting it from somewhere else, extremely well. And then your drug substance. And we're used to small molecules, paracetamol, pretty well defined. But for the drug substance over here, what we used was these CD34 cells that were transduced with the ADA gene. But some of those cells had that transgene in, some didn't. Some had more than one copy in. So how do you define that? And so what we actually came up with was this very, very fancy name that just tips off the tongue. That Strimvelis is an autologous CD34 enriched cell fraction that contains CD34 cells transduced with a retroviral vector that encodes for the human ADA sequence. So that's really interesting. So you think about that as a salesperson going to a clinician and talking to them about it. It'll take the entire call just to tell them what the product is. So these are areas that we really need to understand. We need to understand what that drug product is. What is it that you're putting out on the market? And then also the shelf life. The shelf life of these therapies, as most of you know, is very, very short. And we need to think about, if you want a commercial product, you need to make sure that it gets out to the patient. The patient shouldn't always have to come to you. And so how are you going to ensure that these cells can be cryopreserved and they can be thawed appropriately by potentially people who are not lab trained, who are potentially in an operating room or a transfusion room, that don't have specialist training, but are still going to be thawing your cells and your product and giving it to patients. So think about how you're going to do that. How are you going to make it as user-friendly as possible? Fresh therapies, which is what Strimvelis currently is also very challenging because you have a number of release assays, and very often those release assays haven't read out by the time that you need to infuse the patient and give the patient that therapy. So you need to think about how are you going to do your quality controls in such a way that you are still able to ensure that very high quality for the patient. Within manufacturing also, because particularly for rare diseases, it's very difficult to get hold of patient cells, so very often you're using healthy donor cells. How different are those healthy donor cells from the disease state cells? Some may be relatively same, some may not be. So you need to think about how you utilize those things and how you work with them as you develop your mechanisms and your controls and your tests and your processes. Interesting thing thinking about packaging. Packaging fairly simple. In a clinical trial, you can often just give it in a syringe, or it's relatively simple. When you have to label something that has to be labeled exactly the same, we ran into some challenges here that the medicine or the suspension had to be in a clear plastic bag, and the reason for that was the person that was administering it had to be able to visually inspect it to make sure there was no cell clumping. but you have to have a label attached to it. This is a legal requirement. And so we had to design a special bag with a little flap out on the side that has got that label attached to it. So think about how you're going to administer it. Relatively simple for something like this. Think about if you're developing an inhaled gene therapy or an inhaled cell therapy, how are you going to ensure that the patient gets the full dose? How are you going to ensure that it's appropriately controlled and labeled? So these are some of the things that we ended up thinking about. In terms of regulatory, we can go into this for forever and ever, but the regulatory environment is amazingly getting better, and it's really, really encouraging to see some of the changes, but there are still huge variations. I've already mentioned some variability on the classification of the starting material. The presence of standards, we're still very, very short on many standards, including clinical standards and manufacturing standards, and for that I'm going to give a shameless plug for the standards coordinating body, which is helping to bring together standards from all over the world so that we as a group can develop and use standards that are appropriate to everyone and we can all stand behind. What are the clinical qualifications to treat patients? So are you going to just let any clinician treat these patients, or do they have to be specialists? Do they have to be treated in a specific environment? And then very importantly, just because you have regulatory approval doesn't mean you're going to get your product sold. So think about that as you move on, and we'll talk about that in a moment. So some of the things we thought about, and we talked very long and hard with the regulators, was what are the principles of the active ingredient? What is the active ingredient in this product? Is it the CD34 cells? Is it the transduced CD34 cells? How many of them are there? What volume are they suspended in? And what is their action going to be? How do you define that dose and that strength? And we had some discussions earlier about the dose and about thus the strength that comes from the therapy that you have. How are you going to test that? What are the quality assurance mechanisms that you have in place? What are your quality attributes that you'll use to transfer your manufacturing if you need to? Environmental risk assessment I'll talk about in a moment. Your release criteria, what are those critical attributes that you will test before you release, and when do you release? What is your comparability? If you have to make any changes, what are those attributes that you will consider from comparability perspective? Insertion site analysis, this is critically important. We've had many history with some really, really nasty side effects from using certain vectors in the past. And because we're making permanent changes to these patients, we need to really understand what is happening for these inserting viruses. And so as we look at those, we need to understand what does that insertion site look like, and how effective and how dangerous is it potentially. So we need to understand things like that, and then risk management is critically important. I've mentioned that we have a long-term study, and I'll talk about that in a moment. So I've already mentioned many of the challenges that we faced in terms of non-clinical development, and we addressed many of these with our collaborators at Telethon, but really the reliance on the available non-clinical guidelines was pretty poor at the stage because, as I mentioned, those guidelines weren't really indicative to cell and gene therapies. We had no predictive animal outcomes and no predictive models that really worked very well, and as we've already had some discussions, culturing and using mouse cells is often problematic, and human cells, even in humanized mice, has its limitations. So we need to learn, and we need to become a little bit more imaginative as to ways that we can move that translation from the animal into the human. One of the really, really interesting discussions that we had was with regulators in terms of GMO. So most of you will think about GMO as associated with the food that you eat, grains and things like that. But if you're taking a transgene, putting it into a cell and putting that into a patient, is that patient not a genetically modified organism? And how does a regulator think about that? And so you can imagine, we came in to meet with the EMA, and there was a GMO discussion, and their GMO department walked in, and it was a bunch of farmers and agriculturalists. And they didn't understand any of the words we were using, talking about cells and genes and things like that, and we didn't understand any of the words they were talking about. And so we had to find a way to work together and show them that from our perspective, this is a relatively low risk, and eventually they agreed it's a relatively low risk, but according to the regulations, you will have to go through those both in the clinical studies and also as a final review. And so the big things are to think about: what is the risk of exposure, the risk of the product survival, and the expected waste. Now, if you're giving an injection, it's relatively limited, you can control it, but what about if you have an inhaler? What do you do with that room and all the air in that room after you've dosed the patient? Is there potentially a risk? Do you have to incinerate all of that air, or do you have to demolish the building? So you need to think about some of those challenges that you might potentially face from a GMO perspective. And then in terms of running clinical trials, many of these are fairly similar that you will have seen before. Very small patient populations, particularly with these rare diseases. Finding a placebo group is incredibly challenging because parents don't want to take the risk of putting their children into a randomized controlled trial if there's a chance they won't get a treatment and their child may die. So this becomes very, very challenging. And so the importance of natural history study cannot be underestimated. And I'd urge all of you, if you're looking to start with clinical trials, start with natural history studies, because very often they need to run for much longer and you need a lot more patients in them. The interpatient variability is a real challenge, and this comes down to your starting material. And so you need to be really careful as to what you've got over there. So I've mentioned follow-up, and this is critically important. Once again, as I've mentioned, we're making permanent changes to a patient's genome, and it's critically important that we understand what happens to those patients long term. We, for example, have got a median follow-up of about 8 years now for the SCID patients. But this is a therapy that should theoretically last them the rest of their lives. So what happens at year 10, 20, 50, 60? We just don't know. And so what we've agreed with the regulators was we will put the first 50 patients in for 15 years. And so the last patient will have 15 years in that follow-up. And that ends up to be a little bit over 1,000 patient years to hopefully really understand the long-term efficacy and the long-term safety of these types of therapies. And this is really important. But now, if you start thinking about the logistics of this, if you've got a child that's maybe 2 years old and has this gene therapy and has this change made to them, you are now committing to follow them up for 15 years. And those of you who are parents will know, the child will grow and they'll get to 12 or 13 and they'll become a different species. And they'll stay that different species for another maybe 10 years and then they'll become human again. And so how do you follow them throughout that change and through that time and when they get jobs and go to university? And many of these kids don't even remember being treated initially. And so the last thing they want is some doctor or some clinical researcher coming to them every year or every 2 years and reminding them that they had this dread disease. It's really not cool for your Facebook profile. So we're looking for ways to make this as minimally interventional as possible. And so what we came up with was a way, rather than requiring the patients to go to the clinicians, we through a CRO are going to the patients and allowing the patients to provide that information on a regular basis. So they have an app on their phone and they can use that app to take a photograph of blood results, take a photograph of any lesions or potential adverse events and send them into the CRO, who then has a human analyze all of that and make a call as to if this is an appropriate event that it needs to be referred or what, and ensure that they get followed up, and can then also remind them to provide their information on a regular basis. So we need to think about how we're going to follow these kids up. The other thing is also, it's of limited use having a therapeutic registry. This doesn't really help in the long term. We need disease registries. And so it's important to work with groups that have got disease registries up so that you can take your gene therapy or your cell therapy and compare it to bone marrow transplant, enzyme replacement, and other things and other therapies and modalities that are out there. And this is critically important for us to understand about the differences that we're making to these patients. So hopefully this has given you just a bit of an overview as to how a pharma company thinks about taking these therapies forward and commercializing them. It's not easy, but then again, it wouldn't be so much fun if it was. So thank you very much for your time.